We'll now move from the power grids that keep our cities alive to the biological system that keeps us alive. Our next speaker, Dr. Natasha Latysheva, is a senior research engineer at Google DeepMind, specializing in deep learning for genomics. She holds a PhD in computational biology from the University of Cambridge, and her work focuses on bridging the gap between machine learning and biology. Two worlds that together are reshaping how we understand life at its most fundamental level. Natasha was one of the lead authors of AlphaGenome, a landmark method that predicts how individual genetic variants of mutations in human DNA influence complex biological processes. Her work opens up new possibilities for understanding diseases, improving diagnostics, and driving discovery in modern medicine. She is also passionate about education and accessibility in science, and recently published a book titled Deep Learning for Biology: Harness AI to Solve Real-World Biology Problems, making these complex topics approachable to a broader audience. And for those who enjoy a bit of inspiration, during her time at Cambridge, Natasha once entered the Hack Cambridge, a university-wide hackathon. When her team couldn't agree on what to build, she decided to go solo, competing as a one-person team. Not only did she do the project on her own, she actually won the first prize. Not for the context, Peter was there that year too, and in four-person team they took third place, right? So her story perfectly reflects the spirit of this workshop, combining creativity, determination, and curiosity to push the boundaries of what's possible. Please join me in welcoming Natasha Latysheva with her talk, AI for Biology and Genomics. Natasha, the stage is yours.
Wow, what an introduction. Thank you so much. Yes, thank you for inviting me to this workshop. It's such a pleasure to be here, and also to Montenegro. It's such a lovely country. So today I'll be talking to you about AI for biology and genomics. Just to contextualize myself a little bit more. So I work in the, is there a laser pointer? No. So I work in the Genomics Initiative within the broader science team in Google DeepMind in London. I think this is one of the first pictures you get of the building if you Google search it, and there's lots of clouds, which is accurate. There's no sunshine in London. So the goal of the science team is to use AI to solve some of the world's most challenging scientific problems, humbly put. We're about 200 people across biology, physics, chemistry, maths, and computer science. You maybe have already seen some of the models coming out of the science team before. AlphaFold's probably the most famous one. This won 2/3 of a Nobel Prize in chemistry last year. Alpha missense is another protein-related model that predicts whether mutations in proteins are likely to be disease-causing, so probability of being benign versus pathogenic. Alpha Proteo can design binders for small molecules, so given a target molecule, it will design a protein to bind to that. So that's relevant for drug design and pharmaceuticals. Alpha Proof is a model for verifying and formulating mathematical proofs. Alpha Qubit does quantum error correction, and Alpha Genome is a new model that is from the team that I work in, which I'll spend some of this presentation talking about. Yeah, so this was partially in the introduction, but a bit about my background. In case there's students here, I was also a student for a very long time. So I was originally a biologist. I studied biochemistry at St. Andrews University in Scotland. Then I moved down south to do computational biology at Cambridge, where I worked on cancer genomics. Then I kind of stopped biology for a few years. I was sick of it. I worked in data science and video gaming and in ML engineering for kind of NLP, machine translation. And then for the last 5 years, I've been at DeepMind working on machine learning for genomics. So if you want my career in kind of emojis, it sort of started, solo DNA molecule, then DNA plus kind of computational stuff, then just computational stuff, and now I'm back at the intersection. So for this talk, I thought I would do something a bit different to a lot of my other presentations. So normally I would kind of dive straight into AI for genomics and talk to you about modeling DNA, and then probably spend a lot of time going into all the figures in our recent paper. But actually today what I'm going to do is zoom way out and take you through a lot of different projects in the AI for biology space. kind of sort of rapid fire to give you a sense of some of the work. So actually, I'll spend half-ish of the presentation on that. Maybe I'll cut some of the alpha genome slides, depending on how chatty I get, and then a bit about future direction as well. Okay, so overview, AI for biology. I think it's fair to say AI is growing in popularity across biology, but it's still really early days. Biology itself is incredibly broad, so some of the example projects I'll talk about cover medicine, like medical image diagnostics, drug discovery, and then smaller sides of biology, molecular biology, genetics, genomics, and then also ecology and conservation. So I think it's, to get a sense of these topics, it's just good to see lots of examples of projects, essentially. So let's do that. Right, so cancer detection, this is sort of kind of a classic use case of AI in biology. The ML challenge here is this sort of finding a needle in a haystack problem. So you want to find the signal in noisy, potentially huge amounts of data, and you're guiding really life or death decisions in some cases. This is an example of a skin cancer detection model from MIT. So actually there's two tasks here. One is kind of drawing bounding boxes around skin lesions, so like abnormal bits of skin. And once you have that, then you want to classify each one to, say, predict the probability that it's a melanoma. So melanoma specifically is one of the most commonly diagnosed cancers worldwide. And I mean, early detection is important for all cancers, especially important for melanoma. So it's really great that we have models like this. For skin cancer detection, it's actually quite cool that maybe even 10 years ago, these models were getting comparable accuracy to experts, to expert dermatologists. Of course, it's not the full story. You also need predictions to be interpretable. Clinicians need to trust why the model has made a prediction. In addition to just getting a prediction, then there's also regulatory challenges when you're introducing medical tools, but it's a big part of the way there. On the breast cancer side, this is another incredibly common cancer worldwide. I've chosen to highlight two projects. So we've got LUNET inside MMG. I think this is kind of important because it was part of a massive real-world study across Sweden and Norway. And sort of the headline metric is it showed that doctors can find 20% more cancers than humans alone. Very nice use case of collaboration. MIT's Mirai made headlines a few years ago. So this is a deep learning model that does a bit of kind of forecasting. So it produces a personalized risk score up to 5 years in advance. So by analyzing a patient's mammogram, it can sort of predict the progression of lesions into cancers. And it massively outperforms old school approaches, which is essentially the doctor asking for your age and whether there's breast cancer in the family. So there's a lot of room for improvement, and it's really exciting to see models in this space. Brain-computer interfaces, BCIs, very hot right now. So the ML challenge there is decoding this kind of noisy, massive amount of electrical data that the brain generates and trying to distill that down into either the user's intention or like an image that they're thinking about. Neuralink is one of Elon's projects, controversial figure, but the project is cool. So what they're trying to do is implant this kind of coin-sized chip surgically into the skull with microscopic wires kind of threading directly into the brain's motor cortex. And this chip connects wirelessly via Bluetooth to just a standard laptop. Insert Bluetooth reliability joke here. And one fun anecdote that I liked is that the first recipient of this technology was a paralyzed patient that chose to play video games for the first few hours. So he competitively played Mario Kart and Civilization 6. It's a picture of him there. I'm a big Civ 6 player, so I love this. Another company called Synchron out of New York, I believe, is doing something similar, but they're trying to do it in a way that doesn't require open brain surgery. So what they've got is kind of a mesh-like device that they can push up to the brain through a vein in the neck. And their kind of big headline moment was an ALS patient sending the first direct thought tweet, so not requiring motor input. Sorry, motor output. On the organ and tissue model front, here we're trying to reconstruct a kind of usually 3D anatomical model from data that can be low resolution. It can be from different sort of angles, different sizes of the organ. The SynthSR model from Harvard and UCL did this really nicely, so they can take clinical brain MRI scans, which can be not super high quality, so they can have any contrast, different orientations, different resolutions, and they can turn them into high-resolution 3D images that clinicians can then rotate around and probe and analyze in different ways. DeepMind did a bit of this with retinal diseases. So there's a model that can sort of take these 3D retinal scans and simultaneously do the tissue segmentation, which is in the middle, so kind of going from these gray blobby-like frames into actually labeled structures that clinicians sort of see as more informative. So it's doing the segmentation at the same time as it's doing disease classification. And yeah, the diagnostic performance was really nice. So they classify over 50 different sight-threatening eye conditions simultaneously and outperform human eye experts, expert ophthalmologists. Automated drug discovery. This is such a hot field at the moment. There's a huge number of people doing machine learning for drug discovery, including DeepMind, actually. We've got this pharma spin-out, Isomorphic Labs. The statistic that gets thrown around a lot is that it takes approximately 10 years and $1 billion to bring the average drug to market. So it's an enormous investment, both in terms of money and time. So if there's any way that ML can speed up really any part of this complex pipeline, that would be immensely valuable. I've just highlighted a couple of examples here. One is Recursion. So they're famous for doing really high throughput kind of video-based screening of drug molecules where they've got loads of cell cultures, lots of drug candidates. You drug the cell cultures and you've got cameras hooked up to sort of take videos of cells. So you can read out quickly and accurately how the cells are responding to the drugs. Accenture has the honor of being the first AI designed, of developing the first AI designed drug that actually entered clinical trials. And they did this incredibly quickly. So this, from start to end, was under 12 months, whereas the industry average is 4.5 years. The drug was aimed at treating obsessive compulsive disorder. It did then fail in clinical trials, but it's still really impressive. Yeah, this feeds into some of the previous talks, actually. So on the agriculture side, I thought this is a really interesting use case, crop disease detection via drone or satellite imagery. So the challenge here is how do you process and integrate geospatial data to guide kind of agricultural decision making? There's a company, Taranis, which markets itself as having leaf-level precision. So they use these high-resolution drones to survey fields, and they can detect pests and weeds and disease in the fields. AgroScout does something similar in this field of remote agriculture. I think it's very cool, very sci-fi. Eco-robotics is not a drone, actually. It's a robot that like drives around the field, and it does precision spraying of pesticides. So the idea is that not every plant might need to be treated with pesticide, and on a single plant, not every leaf might need to be treated. So in the end, they can reduce chemical use by huge percents. Biodiversity monitoring. So I really like this project, Rainforest Connection. What they do is they place these modified smartphones in trees and rainforests. They record the audio, and then they use models to identify different species present in the area. So the challenge there is how do you detect species from the noisy audio? And also, how do you build a single map of where these species are from many sensors that are a certain distance apart? So they're using this to monitor biodiversity, and they can also detect illegal activities like logging, because chainsaw audio is quite distinctive in amongst the bat and the bird audio. De-extinction is a bit of a pet interest of mine in the last 2 weeks or so. The main company in this space is Colossal, and they're looking to bring back extinct animals. The way that they're doing that is, so essentially for an extinct species, like a woolly mammoth, they find a living species that's the closest genetic match, in this case, the Asian elephant, and you then want to make targeted edits of the elephant genome to make it more woolly mammoth-like, I suppose. Whether or not that's true de-extinction, I guess, is like a matter of debate. I think it's cool, basically. So the challenges here are, one is genome assembly. I mean, this is a challenging problem and kind of the best of times, classic bioinformatics problem, which is like, how do you build a contiguous genome from small sequences? When ancient genomes become involved, this is even more challenging because the genome can be degraded in different ways. There can be artifacts and so on. And then the other challenge is deciding which edits to make. Like, how do you make an elephant more woolly mammoth-like? Yeah, they had this sort of intermediate result, which is the woolly mouse. So mice are very common model organisms for all sorts of biological work. And they found a gene that's associated with hair growth and hair length. And so now we have the woolly mouse. They're also working on de-extincting the dodo bird. I think they've already got the dire wolf de-extincted. And we also have the thylacine. There's a funny section on their website where they describe how this is not Jurassic Park. Check that out. Basically, yeah, dinosaurs are too old and DNA half-life is too short. So yeah, actually, a bit more about genetic engineering. So CRISPR technology is really amazing, but it's not perfect. One of its main weaknesses is what's called off-target effects. So you can design a system that will make, you hope, precise edits to somewhere in the genome, but odds are it's going to make cuts elsewhere, sometimes in really bad places that can cause cancer. So there's a few deep learning kind of projects and companies actually that are trying to predict and prevent these off-target effects. And AI is also finding entirely new tools, CRISPR-related tools that humans have missed. Broad Institute has found hundreds of naturally occurring novel Cas families related to Cas9, but different, by sort of trolling through many bacterial genomes using metagenomic techniques. And other research have found sort of smaller scissors, enzymes that can edit RNA as well as DNA. Yeah, lots of like kind of data mining work has been really fruitful in this space. I love to stand by all these weird animals. Okay, so yeah, this is a problem that I hadn't really thought of much until I became involved in a sea turtle face recognition challenge. But basically the same way that we do facial recognition for people, animals' faces are also distinctive, even if humans can't pick up on it very easily. So for example, this is a grid of lemur face images, and these are different lemur individuals, right? They look the same to us, but not to other lemurs, presumably, and not to machine learning. So there's a few projects here. Bear ID project is monitoring individual grizzly bears in the wild. That's quite cool because bears change weight dramatically due to hibernation cycles. They also get wet. The algorithms have to be robust to that. Lemur Face ID is here. This is tracking highly endangered lemurs in Madagascar. And Chimp Face is scanning social media to find photos of captive chimps in order to combat kind of poaching and animal trafficking. Animal speech decoding. This is like Dr. Dolittle stuff. So animals, we know, have complex communication systems, but we can't talk to them. They don't speak human languages, but they do speak some sort of language. So Project SETI is one project that's working on decoding whale sounds and understanding their communication and behavior. Earth Species Project, also looking at whales as well as bats. And I think they've actually made some progress, and they found out the names of some whale individuals, which is quite cool. Antibiotic-resistant models. Petar actually mentioned the Halicin project. This is a project that found a new antibiotic drug that kills resistant superbugs that have become immune to other antibiotics. DeepArg is a deep learning tool that reads bacterial DNA and predicts which specific antibiotics they would be resistant to. Viral evolution forecasting. This has become more pressing after COVID. So EVESCAPE is a tool that forecasts which SARS-CoV-2 variants could dodge innate human immunity by analyzing protein structures. NextStrain is not strictly machine learning, really, but it's more like a data collecting project that's moving into forecasting. This is an open source project that uses genetic data and sort of tracks viral mutations globally across many different countries in real time. It sort of pitches itself as a Google Maps for flu and COVID spread. And folding proteins, of course. So when I was studying biology as an undergraduate, this was pitched to me as the holy grail of molecular biology. So this is the problem of predicting how a protein folds in three-dimensional space from its linear sequence of amino acids. And yeah, I mean, the way the lecturer was talking about the problem, it seemed basically impossible. But yeah, it was basically solved, and John Jumper, Demis Hassabis, and David Baker got this Nobel Prize last year. Here you have the structures overlapped, the experimental results, with the computational prediction. Not unrelated to folding proteins, you have designing new proteins. The results I wanted to highlight here, there's a new startup, Evolutionary Scale, by former Meta researchers, I believe. They were looking at GFP, green fluorescent protein. GFP is like a very standard, important tool in laboratory biology. The idea is that if you're studying a specific protein, you can kind of tag this fluorescent tag onto it, and then you can track it under a microscope. You can see where it is within the cell. So what they did is they used their protein language model, ASM3, to design a new fluorescent protein that is quite distinct from the ones that we have in nature. The quote that they have is that their model is simulating 500 million years of evolution. I think that's just computed given the rate at which GFPs evolve in nature, and then looking at kind of the sequence diversity between their proteins and existing ones. Yeah, designing viral genomes. The main player that comes to mind in this space is Arc Institute. So they've got these two papers, EVO and EVO2, on genome language modeling. One of the things they showed in the EVO2 paper is this bacteriophage design problem. So I mentioned earlier that antibiotic resistance is a really big problem for medicine. Well, there's another way to attack bacteria without using antibiotics, and that's using bacteriophages. So these are essentially viruses that are the natural predators of bacteria, and there's many, many of them in nature. What they did in the paper is they took the genome of one phage, FX174, and they iterated on that genome to see if they could improve its ability to attack the bacteria. And it happened. Some of the variants actually attacked the bacteria much better than the original wild-type phage. Yeah, designing bacterial genomes, I think it's not controversial to say that this is a harder problem than designing viral genomes. Bacterial genomes are much bigger, and they're independent organisms. They have to do all of their own metabolism and replication, unlike viruses, which use host resources. The J. Craig Venter Institute is quite famous for this minimal genome project. So they didn't kind of start from scratch. They started with the bacterial genome and kept chopping away at genes and seeing at what point, like how minimal can you go before life disappears. The end result is a very small genome, which is 500,000 bases, less than 500 genes, and it's the smallest genome of any self-replicating organism. This was actually sort of 10 years ago, so it's, I'm not sure how much ML was used in that, but nowadays in synthetic biology it's quite ML heavy. Yeah, one final thing I wanted to talk about, which I think is kind of the future, is AI for accelerating science workflows themselves. So it's not specific to biology specifically, but basically, how can we use AI to do things like automate our analyses, do the data cleaning, do the data munging, generating literature reviews. LLMs kind of do that already. What about linking concepts together? What about hypothesis generation? And probably in the not so distant future, we might see fully automated kind of lab systems where an AI scientist can generate a hypothesis, the roboticized lab can do the experiments, get results, the AI can interpret the results and sort of iterate on its own. There's very recently been seemingly a lot of work in this space. So first I'll talk about DeepMind co-scientists. On the upper right, the idea here is that the scientist specifies a research goal to the AI co-scientist. This can be super long, like super detailed prompts, including lots of really specific science. Then there's kind of a series of Gemini agents. Gemini is Google's LLM. So you can have like an agent that's generating ideas, one that's ranking them, one that's reviewing them. And in the end, what happens is that there's this research ideas tournament where different ideas or hypotheses kind of fight each other and get ELO rankings. And the scientist sort of gets like the top 10. Very recently, Claude was released, Claude for Life Sciences, anyway, from Anthropic. I think this might have been 1 or 2 weeks ago, actually. So they're focusing on kind of automating bioinformatics workflows involving anything relating to genomics, actually, so like RNA sequencing, for example. And then very recently, I want to say maybe Thursday, tells you when I made these slides, Cosmos from Future House was released. This is another AI scientist, and they're saying it's made 7 kind of major scientific discoveries so far. I think this is a very interesting trend. Also, actually, I like this trend of kind of tech companies doing these high production author chats, like kind of fireside roundtable chats. Yeah, I really like watching them, so recommend them. Yeah, so that's kind of all I wanted to say for this survey. Let me know if you have any questions, just feel free to jump in. Otherwise, I'll head into what I actually work on, which is AI for genomics. Okay, so why are we modeling DNA? Why do we want to use machine learning in genomics? So I think we don't have any evidence to the contrary that DNA is the basis of all life as we know it. It contains all the information on how to grow an entire organism from a single cell. It also controls how cells should respond to stimuli, how cells grow and divide and transform into tissues and organs of various sorts. And it also contains the key to how diseases work. That's not to say DNA is the full picture. There's also, epigenetics. There's also the environment of the entire planet. But still, understanding DNA is, I would say, foundational. It's a root node problem. If you really understood how DNA worked, this opens up a huge amount of use cases across biotechnology, pharmaceuticals, medicine spaces, and so on. Yeah, so even though we've had the human genome sequenced, the reference human genome sequence since 2003, yeah, I would say we're really still at the beginning stages of figuring out what's in this massive bookcase of 3.2 billion letters. So I feel like we're at the phase where we know the what, like here are the letters, but not sort of the why or the how, or at least we're at the beginning stages. There's this famous quote by Eric Lander. This was made right after the completion of the Human Genome Project. Genome bought the book, hard to read. And yeah, 20 years later, I think we're still at that stage of confusion, essentially. But I think there's promise because deep learning is very powerful for dealing with huge amounts of data. And in this case, it's kind of text data, right? It's 4 different letters in a book. So the thing that my team and I focus on at DeepMind is understanding the kind of molecular function of DNA. So we're not trying to predict, like how is an entire organism going to develop from a single cell. Like we want to understand at a molecular level, you have this bit of DNA, what's happening locally around it. So to give an example, here's a long string of DNA in the cell nucleus. And if you sort of zoom in, you see some interesting molecular biology. So you see a gene in green. We want to be able to say, like, in this string of DNA, this is where the gene is, this is the structure of the gene. It's expressed in high levels in liver tissue, but not in brain tissue. The context that it's in is kind of open. It's not compacted DNA. And maybe like 3 different proteins bind that contribute to the regulation of that DNA. So this is kind of what I mean by saying we're modeling the molecular biology of the DNA. And yeah, of course, proteins are very important, but 98% of the genome is actually not coding for proteins. So it's non-coding regions. And most of the variants that we know that are associated with diseases are in these non-coding regions. And there's many possible mechanisms by which those variants lead to the disease, but generally we don't know them. Like there's very little that we understand about those mechanisms. Yeah, so this is where our work comes in. So we work in the space of sequence-to-function AI models. Essentially, we take in a DNA sequence. So by 1 megabase, I mean 1 million different DNA letters, ACs, Gs, and Ts. And we want to do this kind of sequence-to-sequence prediction problem where, given the DNA sequence, we're predicting readouts of the experiment that characterizes that DNA's function. So here, we're saying that those peaks correspond to where genes are expressed, for example. And if we can do this, then if we can do this well, then we should also be able to generalize to perturbations of the sequence. So if we change a single letter in the DNA and rerun the predictions, how do the model's predictions change as a result of that single mutation? Right. So why do we care about this, I guess? How many people have heard of GWAS before, genome-wide association studies? Nice, a few. So GWAS is basically our best tool for linking mutations with some sort of effect, like a disease or height or BMI, some sort of phenotype. And we have these databases that say that, you know, at this position on chromosome 11, if you have the letter A, you're more likely to be healthy. Whereas if you have the letter T, you're more likely to have the disease. And you can go and look at these databases. On the right, this is a chromosome showing all the different GWAS hits at each position. And you can see just how many statistical associations we found. And the hits keep going up, basically. But essentially, for the vast, vast majority of these hits, we really don't know why there's a link between the DNA and that phenotype. We just know that it exists. And yeah, I think if we can build better models of DNA function, we'll be able to answer why these links exist, essentially. So there's a lot of information encoded in the DNA level, which kind of goes to the RNA level, to the protein level. Biology is very interconnected anyway. So yeah, basically we hope that by modeling some of these processes, we can make kind of causal links between mutations and phenotypes, like diseases. This is a graph of some of the supervised DNA sequence deep learning models. So we're not the only people working in the space. This is a couple of years outdated now. But essentially on the x-axis, we have different models, different genomics, deep learning models. And on this axis, we have the type of model output. So what is the model predicting from the DNA sequence? Is it predicting DNA features, like the activity of genes and how compact the DNA is? Or is it predicting RNA features, such as the stability of the molecule and so on? So one thing you can see is that some models kind of hyper-specialize into predicting one thing, whereas other models kind of try and do several tasks at the same time. So I would call these multimodal models. Yeah, and if you look a bit more into the space of existing models, you see that there's some trade-offs that start to emerge. So I think the most fundamental trade-off is between the length of the input sequence that we're modeling, so the length of the DNA, and how fine-grained the model's predictions are. So in general, you want the input context to be as long as possible because it can be really biologically important. Like to predict the activity of a gene over here, you might need to know a bit of sequence that's 500,000 bases away. So very important long-range regulation can and does happen frequently. You also want the model's predictions to be as finely detailed as possible. A lot of interesting biology really does happen at like one base pair resolution, like splice sites, for example. But essentially, it's just difficult compute-wise, it's difficult memory-wise. Yeah, so different models kind of occupy different parts of this trade-off space. So we have models like Informer, also from DeepMind, and Borzoi from Calico, David Kelly's group, which have gone kind of long input, but low resolution. We have other models that go very high resolution but short input length, models like Splice AI from Illumina. And then kind of separate, slightly separate axis is, do you go full on in one modality? Like, do you hyper specialize into, say, splicing or like how DNA folds in the nucleus, versus do you try and model multiple things at the same time? Oh yeah, and then like these hyper specialized models do tend to be state of the art, but it's just challenging. Like if you're trying to analyze the effect of a mutation from many different lenses, many different facets of molecular biology, then you're having to sort of use multiple models at the same time, and it's just not really convenient or easy. So this is where our model comes in. So we have longest input DNA sequence yet. We take 1 million letters. We predict at the highest possible resolution for most of the outputs. And we try and kind of pack as much biology into a single model at the same time as possible. So many different modalities under one hood. And a lot of them are kind of related anyway, so we don't get degradation on individual tasks as we kind of keep adding more and more. A bit more about how the model works. So it's trained on all of this, like these functional tracks that I've been showing you, these like readouts along the DNA sequence. And luckily, there's already huge data consortia that have gathered a lot of this data. We owe a huge debt to people that actually do laboratory experiments and clean up the data. I mean, we still had to clean it a lot, but it exists, and yeah, we're eternally grateful. So because the input sequence is so long, we had to do some sequence partitioning across devices, make sure they communicate efficiently. But essentially, yeah, this is the input and output. The input is just DNA sequence, and we're predicting like thousands of tracks from that. So we're predicting all about like the expression of genes in that region, how packed the DNA is, protein binding, and so on. And all of these are tissue specific, so the signal in like heart tissue will be very different from signal in liver tissue, and we want to capture those differences as well as possible. Yeah, so we kind of have two phases of training. The pre-training phase is by far the most important. This is what we actually did on the previous slide. So we train using observed experimental data and evaluate on held out genomic regions that the model never saw during training. We then also have this distillation procedure. This is kind of to squeeze a bit more juice out of the model. It's a bit like taking like a mean ensemble over different like seed trained models, but without the heavy inference cost. And this is what the output looks like. So in this diagram, we have pairs of observed versus predicted tracks. And this is kind of a zoomed out high level view. And that's just to show that, you know, we're roughly predicting the right shapes of these signals. We can zoom in to a region there, and you can see some more interesting things. So keep in mind, the model has no concept of genes or gene structure. But if you then overlay that information, you can see that it's recapitulated it very well. So there we have a plot of the LDLR transcript, and you can see that the model has learned like with the intron and exon structure of the gene really nicely. Right, evaluations. If you do happen to look at this paper, it's extremely long, and it's just absolutely like packed to the gills with evaluations. We just wanted to, you know, really do justice to all the different models we're comparing against, and also all these different modalities that we've sort of shoved in. But yeah, basically on like track prediction, we do reasonably well, improve over the best existing state-of-the-art model, even specialized single task models. So we were really pleased to see that. And yeah, actually, I realize it's starting to get a bit detailed on this paper, so I thought maybe it's a good time for an interlude. How many people here read Hacker News? Okay, that's quite a lot. Yeah, I see Hacker News as a bit like Reddit, which is, you know, it's mostly like a place to waste your time and have fun, but occasionally you do come across something really, really useful. And this happened to me. I came across something really useful about 2 years ago, and it was this post. So it was Ask Hacker News, what's the most important problem in the world, and are you working on it? In the comments, it turns out this is related to the Hamming questions. This is an anecdote about a mathematician, Richard Hamming. So yeah, I can read it to you. Mathematician Richard Hamming used to ask scientists in other fields, what are the most important problems in your field? Partly so he could troll them by asking, why aren't you working on them? And partly because getting asked this question is really useful for focusing people's attention on what matters. So, yeah, I don't know, that really stuck with me. I was reflecting on it in the context of our work. And in genetics and in genomics, I actually do think that this question of variant effect prediction is the most important one in the field. So just to recap, it can be described very easily. So basically, given a DNA string, a model makes a prediction. And then we introduce a mutation, we make another prediction. And what are the differences between those two predictions? So it's kind of simple to describe, but it's an intensely difficult and thorny problem. The reason I think it's so important is because the consequences are huge. If you can really start to understand why specific mutations are linked to effects, yeah, so many things open up, like those GWAS studies I was mentioning earlier. But yeah, in order to do that well, you need a good model of DNA function to be able to do this variant effect prediction. And yeah, so with this Hamming question, the way I kind of choose to interpret it is a bit more personal than this formulation. I would say, like, what do you think are the most important problems in your field? Like, I don't think that people would necessarily have the same answer to that, but I still think it's a good question to kind of prompt you to reflect on what you think is most exciting, and perhaps should you perhaps work on that. Back to variant effect prediction. So this is what the output of the model actually looks like. So yeah, along DNA sequence, we predict these signals. I told you that we have sort of the mutated form of the DNA and the not mutated form. This is what's meant by ref and alt. So we predict these overlapping lines. And you can see that when you introduce the mutation in red, the gene's expression signal plummets. So we lose expression of that gene in the tissue, which is colon tissue. And we can do this type of variant effect scoring for all the different modalities we've included in the model. Essentially, we're always comparing these red and blue lines to see what the differences are. This is kind of a visual view of variant effect scoring, but for our evaluations, usually we need like one single score. So then like variance scoring is all about summarizing those differences in various ways to produce a single number. This took a lot of time to work out, but yeah, it's just a way of kind of aggregating these differences between the red and blue lines. And this was probably the most important figure of the paper. It's not very like pretty, but basically we're showing that compared to all the different genomics models in the field at the moment, for each modality, we're generally better at doing variant effect prediction, except for one, the pangolin row, that caused a tense meeting when we discovered that number, but it's okay, we can't be perfect. And we were really happy to see that our massive kind of multimodal, super multitask model is outperforming really specialized single task models. I think that was really nice validation that you can have something like a foundational model of biology. You don't have to hyper specialize into just one task. Yeah, so this, actually, what time is it? Oh, I'm okay. I'm doing okay in time. Yeah, so then I'll go into this case study. So the idea here is, okay, so this model seems good at different types of biology. Can we use it to recapitulate some known experimental data, which would kind of give you confidence that in the future, maybe you wouldn't need to do so many experiments and you could just use the model to predict the effects of mutations. So, so we have a region of the genome here on chromosome 1. There's a gene called TAL1. Genes are kind of not, like protein coding regions are not contiguous in the genome. They have to be cut and spliced together. That's what these different boxes are. The big boxes are the exons. And we have 3 different papers that have studied this region in green and purple and blue. And the yellow triangles are different mutations found in those papers. And these papers have done very complex biology to understand what each of these mutations does to the gene and how it's involved in this cancer, which is a type of leukemia. So what we want to do is we want to use the model to see what it thinks about what these mutations are doing. So that's what we did. We collected all these different mutations, and we kind of ran them through the model and see what came out. So what we saw is that the true cancer-causing mutations, if you compare them to kind of random mutations also in the region, they really jump out. So they have a much higher predicted change on the gene in the relevant tissue compared to kind of matched controls. And you see that for all the different types of mutations, single letter changes, like multi-letter insertions, and I think also deletions. So yeah, maybe I need to hype this up more. We were so happy to see this, actually. So yeah, just to recap, the model has never seen this region before, but it can look at these cancer-causing mutations and really highlights them. It really says, like, yes, we think this is definitely involved in changing the expression of this gene, which we know is related to this cancer. And we can do this across different modalities. So this plot was just on the expression side. But if you kind of score all these cancer-causing mutations across our different outputs and cluster them, you see that the cancer-associated mutations in yellow here form a really distinctive cluster compared to the controls. So there's really something happening here. The model seems confident of that. Yeah, we had lots and lots of kind of variant case studies in the paper. Feel free to look. But yeah, maybe just one more example. So here we have a mutation. So on chromosome 11 at position 11 million or something, we're changing a single letter T to a G. And the problem is predicting the effect of that. And it looks like the model is saying that it's reducing the expression of this one ApoA1 gene in heart tissue. So these are very kind of precise problems in a way. These are mildly cherry-picked. We also have examples where the model did not do well. But I think it's nice that in a lot of cases it gets it right. And yeah, ablations, I will not go into these plots, but I think we learned from AlphaFold, they had a lot of feedback that people really appreciated the ablation figure. And of course, Google is this kind of behemoth with lots of compute. Not everybody can do this type of analysis, so we really poured a lot into ablations. And by that, I mean we're chopping away parts of the model or parts of the data and seeing how this affects the results that we care about, seeing what's really crucial for the performance versus what's kind of fluff. Yeah, just a bit about the limitations of the model. So yeah, I mentioned a bit about long-range interactions. I mean, I think this is just a really complex problem in machine learning in general. The problem is that biology also has this distance bias. So yes, distal things can be important, but generally close things are much more important. So it's kind of compounding, like ML algorithms struggle with distal information, and then biology doesn't really pay that much attention to distal elements. So where there are important distal elements, these can get missed. The model also struggles a bit with tissue specificity. So we do predict all these signals for all like hundreds of different tissues, but there is a slight regression to the mean effect. This also happens with populations. If I try and predict the gene expression of everyone here, the model will do a good job to predict sort of the median gene expression, but kind of deviations on either side is much more challenging. Species coverage is still quite limited. We did explore expanding species considerably, but yeah, it's challenging to know how to make use of species that are so diverged from human. Mouse is still helpful. So if you cut out mouse data, human performance gets worse. But if you start adding in more distant species, yeah, it kind of falls over a bit. So at the moment, we only have human and mouse in the model. Yeah, I mentioned personal genome prediction. All of this work, and actually maybe like 90% of work in genomics is still on the reference genome. Shamefully, I think as a field we'll start to move more towards personal genome prediction. Yeah, and the fourth one, I mean, it's kind of a limitation of all these models, but basically if you're trying to do analysis of traits involving like broader biological processes, like if you're studying a disease, this is kind of beyond the model's molecular scope. Like a disease depends on so many things. Yes, DNA is a huge part of that equation, but also things like, diet and carcinogens and so on. Yeah, so that's all I wanted to say about AlphaGenome. A quick section on future directions that I think are particularly interesting. So what are the major challenges for AI in genomics at the moment? Probably not a surprise that the first point is data. So at the beginning of the talk, I had this slide, where the genome is like this bookshelf filled with all these books, and it looks huge. I mean, it's true, but in a way, the genome is not actually that large, and there's limited training data if you're only looking at human. So there's only 20,000 protein coding genes in the genome. And there's also repetitive regions. So at the ends of chromosomes, at telomeres, and in the center of chromosomes, at centromeres, these are highly repetitive regions that are just not really that interesting. And we don't really use individual genome data well. We train on the reference genome. So yeah, there's also relatively little experimental data for other species. As you go further from mouse, it really drops off a cliff. So if you're training these types of big supervised models, you're just limited by the experimental data that's out there. So yeah, I mean, DNA language models are a thing for sure. I think they're just now starting to be really impressive. For a few years, definitely lagging behind a lot on the type of tasks that people actually care about downstream. But they don't have this problem, right? Like they just need genome sequences to train on. They don't need label data. So maybe we'll see them take over a bit in the next few years. Also, yeah, benchmarking in genomics is really tricky. It's not really mature. So bit of an anecdote. So we in genomics actually sit very close to the protein team, the AlphaFold team. And occasionally we get a bit jealous because their problem is so well defined, like they have the ground truth right there. If you predict a protein structure, you can overlay it and see like how good are you, like the notion of what's good is really clear. Genomics is really more still in this regime of fragmented evals. There's no single measure of what's good or not, and different papers kind of come up with their own benchmarks. There's some kind of community aggregation, but not that much. It's just a fuzzier field in general. And I think also figuring out how to combine these different kind of levels of information processing, the DNA level, RNA level, and protein level, this will surely become important, right? Like it's a bit weird that these models are separate at the moment. But yeah, I mean, we're still very early in the game. For protein structure, first structure was maybe in the 1950s. First genome was in 2003. The first complete human genome was in 2022, the telomere to telomere genome. And yeah, I mean, cost of sequences of sequencing really has plummeted, but it's, I don't know, it's not clear how to use this data. It feels like really early game at the moment. Right, and yeah, one thing I kind of want to highlight, we've been talking about DNA as this sort of linear view, like you're reading a book. The reality is something far more chaotic. So this is, these are different chromosomes in the nucleus. They're sort of segregated into different chromosome territories, which are quite stable actually, but the idea is that they're tangled up with each other, and the organization in 3D space is actually really important. And current models do not really take that into account that much. Yeah, and then time. Time and space. So the cell is such a busy and crowded place. The levels of molecular crowding are just unbelievable. The models that we have now are so abstract. They don't really have a notion of time or space. Time because, yeah, these experiments are so expensive, people are not going to do time series of them. Hopefully that'll change. And space, yeah, maybe just historical, really. I think we'll definitely see more models explore these concepts. It's a pretty GIF. We can watch it for a bit longer. Cool. Yeah, and I just wanted to finish off on some data projects. There's really been like an explosion in the last few years of sometimes really large-scale genomics data projects. One that was quite recent, I think this year, is the Emirati Genome Project. I think this is a really good example of a government going all in on genomics and just deciding like, we're doing it. So they want to integrate genomics workflows into standard healthcare for everybody, for every either resident or citizen. But they're not releasing it though, like they're quite guarded. The Earth Biogenome Project is trying to sequence the DNA of all 1.8 million known species on Earth within the next 10 years. The idea being is that we are killing species off very quickly, and we need some sort of like backup we need their genome before they go extinct. It's a bit dark, but it's better to have that than nothing. The human pangenome, as I mentioned, genomics has been largely studying the reference genome. This is actually mostly the genome of one guy from New York. It's not quite that, like it's a bit of a mosaic of a few people, but it's mostly one guy. So this project is trying to replace that with sort of a map from various people of diverse ancestries. Then there's the All of Us project by the National Institute of Health in the US. So they've released 250,000 clinical grade genome sequences, half of which are from underrepresented minorities, and have found like just a huge amount of genetic variants that we didn't know about before. UK Biobank is super famous. So they have 500,000 whole genomes along with decades of electronic health records. And this dropped fairly recently, and yeah, it's just like mind-boggling, like to imagine the amount of information that's in there and like where do you even start to analyze it. I did want to mention that the UK is actually really good for genomics globally. So NHS is currently sequencing 100,000 newborn babies. And if that pilot goes well, they want to then start sequencing everybody that's born in the UK. I think that's really cool. But again, intimidating, like the amount of data. But yeah, we'll figure it out. And finally, Human Cell Atlas is this complete map of all human cell types together with single cell sequencing. So we're not sequencing like a bulk cocktail of a tissue sample. We're actually going in and sequencing individual cells. And they're doing that across different tissues, across ages, and across disease states. Okay, I think that's nearly all I wanted to say, really. When I was making the slide, a colleague walked by and said to not use this image because it looked evil. But does it look evil? I don't know. It's a little bit. Google is not evil. Yeah, anyway, I just thought it was look kind of cool in sci-fi. So to summarize, we're building models of DNA sequences. We think this is foundational for understanding biology. It's foundational for understanding health and disease. So supervised models like ours are trained to predict these experimental readouts of DNA properties from the sequence. And these are things that capture things like gene expression levels and protein binding to DNA. We then use these models to predict the effects of variants. We hope this will kind of elucidate all these missing links that we have between mutations and the diseases they cause. Hopefully we'll unpack some of those mysteries there. And yeah, later I would like to see better integration of DNA, RNA, and protein biology. It'd be nice to have a proper foundation model of the cell. Eventually, we'll also need to incorporate notions of space and time into these models. And yeah, genomics data is absolutely blowing up. So how can we best make use of it? And at the same time, certain kinds of data are really lagging, like these supervised signals. So how can we get more? How can we encourage the community to do more of the experiments that would improve these models further? So yeah, I think it's a really great time to get involved in this field, in biology generally and in genomics specifically. As you saw in the first bit of this talk, in biology, there's so many different areas to explore. And I think a lot of them are really just getting started with ML, to be honest. So there's plenty of space to sort of get creative and pitch projects, both academically and in the startup space as well. Presumably, I'm not in a startup, but seems good. And in genomics, yeah, people are really just now starting to build really impressive models. And my hot take, my like mild one chili pepper hot take, is that genomics is not really bottlenecked by compute. Some of the most interesting papers I've seen in the last year or two are doing analysis work to try and understand what existing models have learned, understand their strengths and weaknesses, applying them to new problems, and so on. Yeah, learning resources in this field. Yeah, I'm a bit allergic to like self-promotion, but we did write a book on this topic if you're interested. The reason we wrote it is because there's really not that much at this specific intersection. So yeah, hopefully it's useful to someone. And Petter actually reviewed the GraphNet chapter, so thank you so much for that. What a nice guy. That's all for me, thank you.
Thank you, Natasha. So do we, yeah, we do.
First of all, thank you for a great presentation. It was really insightful. So I'll get back to the first part of your presentation about AI in biology. So you mentioned a lot of examples which were really interesting. So where do you see which like top 3 examples do you see as a first in commercialization? And the second part of the question is maybe more interesting for the others is where do you see investors pouring most of the money? Thanks.
Yeah, great question. I feel like we could spend an hour just talking about that. So look, I think the medical imaging side is really exciting. I also think healthcare around the world is extremely reactive. We don't really assess people for risk well enough. We don't take scans and check for early signs. I think that's something that AI can help with immensely. There is sort of some resistance from medical professionals, like they don't want to feel like they're being automated away. I don't think that's what these tools are trying to do. It's about improving their ability and helping them do work faster. So medical imaging, I think, is really good. I'm a bit skeptical about drug discovery. I feel like, I don't know, there's like a new drug discovery startup every week. Like they can't all be like discovering interesting new things. I guess some must be. I hope de-extinction works. That's really awesome. And we've seen like a huge amount of success on the molecular biology side, like protein folding, protein design is going really well. Investor side, presumably drug discovery. That's so all these startups are being funded. Yeah.
So I have like 2 comments and 1 question. Just short remark regarding cost of drug development. You have stated that is 1 billion. Costs are actually skyrocketing, and as of this moment, 1 drug costs $3 billion to be developed, risk adjusted more than $10 billion. So we can discuss this later. My second question, and kind of a comment, do you know why they have chosen those rare diseases for evaluation of genome impact on outcomes? So you mentioned that they were like really rare leukemia. Why they choose exactly this leukemia and not some other rare disease? So, and I have comment upon of it, please.
Yeah, good shout on the 3 billion, 10 billion. I don't know what risk-adjusted cost means, but we can talk later. Right. Why did we choose that example? It was actually a personal thing. Like one of the team members worked in the lab where they were studying this locus, so he just happened to know of this gene and wanted to look at it.
So, and my comment on this, because I have previously discussed one other kind of a problem with Petar and with Razani. So nobody have really a simple big picture. How many diseases is out there, how prevalent they are, how many times are they mentioned in literature, and with which kind of other entities those diseases are connected. So this is also one of the very basic and apparently simple problem that nobody have addressed.
Yeah, I'm actually interested in that, like pan-disease models, I think are really cool. You're right.
And we can discuss this a little bit later. Thank you.
Okay, so thank you very much. That will be all for the questions. If you want to ask Natasha something more, then you can do it on the hashtag chat channel on the Slack or to approach her